Hi, everyone, and welcome to, I believe, the fourth in the series of Orion Bionetworks webinar series. Today we are going to welcome Lee Lancashire, PhD. He's a bioinformatician in the computational biology group at Thomson Reuters. Lee's work focuses on the identification and development of strategies for the delivery of informative biomarker signatures from big omics data through the use of sophisticated statistical algorithms in combination with Thomson Reuters' knowledge base of pathways and interaction networks. Lee's the project manager for Thomson Reuters' collaboration with Orion Bionetworks, a global alliance of leading organizations in patient care, computational modeling, translational research, and patient advocacy. He concentrates on data analysis with the aim of developing predictive disease models, identification of novel drug targets, and drug repositioning to advance the development of new multiple sclerosis treatments. Lee has a PhD in bioinformatics from the Nottingham Trent University. He's an author on over 25 peer-reviewed scientific publications, and he's the inventor on 4 US and European patents. He completed his postdoctoral research in biostatistics and bioinformatics at the Patterson Institute for Cancer Research, and he's also worked as a bioinformatics team leader in personalized medicine and diagnostics at CompanDX and Almac Diagnostics. I also have been led to believe that he is a former astronaut, professional rugby player, Olympic gold medalist, and multiple Oscar winner. But I have not yet confirmed that information. But ladies and gentlemen, today's topic is pathway and network-based discovery of gene signatures. Please welcome Lee Lancashire.
Thanks, Lenny. And yeah, before I start, I can indeed confirm all of those things you just said about me. So,
thank you.
Okay, so I think we'll jump straight into the presentation because there's quite a lot to cover today. So I'll just share my screen with everybody. So hopefully everybody can see my screen now. So yeah, thanks for the introduction again. So today I'm going to be talking about the identification of biomarker signatures from high throughput molecular data, such as those generated by omics type analyses. So when I say omics, I mean transcriptomics, gene expression, genomics, proteomics, those type of data. And I'll give a general introduction to the concepts of how we begin to analyze these data, some of the concepts regarding the statistical approaches that we take, and then I want to describe several examples from the literature and also from our own research as to how we might incorporate network and pathway information to identify these gene signatures. So this is just an outline of the presentation today. So as I said, there's quite a lot to cover during this hour. So I'll begin by introducing the concept of biomarker identification, and then I'll introduce some methods which are popular for data exploration. I'll go over some concepts of classification and show examples from the literature of some successful gene-level biomarker discovery. Then I'd like to talk about how we integrate pathway and network information to help us further with this biomarker discovery process. And then I'll finish with a real-world example where we've used our proprietary database of protein-protein interactions, known as Metabase, in identification of a time-to-relapse signature in multiple sclerosis as part of the Orion Bionetworks program that Lenny just introduced to you. So I'd just like to start off with the pure basics, so just to define what a biomarker is so that everybody's on the same page. So here you can see the definition of a biomarker according to the NCI, and it's as follows. So it's a biological molecule which we can find in blood or any other bodily fluid or tissue, which is indicative of any normal or abnormal process or some disease. So we may use this biomarker to see how well the body responds to a treatment or a condition. And we also refer to biomarkers as signatures in some cases.
Lee, this is Jeremy.
Yeah.
Is it possible to show the slides in slideshow mode and they're not advancing on the screen here? There we go.
Oh, so I was in slideshow mode. So it's easier if I stick to this preview like this?
If you hit set up in the slideshow, 2 icons to the right.
So I had, can you see that now?
It's a little small.
Is that advancing or not?
I see you're on slide 3.
Okay, so it looks like when I go to slideshow mode, you'll not see my slides advance. Tell you what, let me reshare. Sorry, just bear with me a second. It looks like something's blocking it from sharing it. Okay, so do you see this?
Yes.
And is it advancing?
No, it did not advance for me.
Oh, okay. Let's try. How about now?
There we go. Yep, perfect.
That's advancing?
That's it. That's perfect. Thank you.
Okay.
Sorry to interrupt.
Yeah, well, thanks for telling me. Otherwise, we'd have been stuck on the title for the rest of the talk. So, yeah, let me continue. So, I've defined what a biomarker is. So, this slide just talks a little bit more about those. So, I think we all appreciate that most drugs don't work in most patients. So, a relatively small proportion of drugs actually go on to become approved. This was around 10%. So with this in mind, it's understandable that pharmaceutical companies want to lower the risk that they actually take on which drugs they progress. So one solution is to start to think about how we separate those patients who will respond well to particular therapeutics and those who show some resistance. So this is known as patient stratification, and biomarkers may help us with this. So to identify these biomarkers, we think that we need a combination of knowledge, of content, of expertise, and experience. So biomarkers are a pretty hot topic at the moment, and clinical biomarkers have shown a rapid growth rate over the last couple of years. And they can be used to guide decisions in the clinic. So as I said, they can help us predict drug response, they can help us monitor toxicities, they can help us with early diagnosis of disease and also patient stratification. And as a company, Thomson Reuters, we have various data assets. We have the tools and the expertise to help perform these types of biomarker discovery programs. And we're focusing particularly on these omics type data analysis for these studies. So we base our approaches on integration of advanced statistical techniques, on network biology and extensive biological pathway information. So I'll talk more about these approaches during the presentation. And we've applied these approaches for many major pharmaceutical companies in biomarker discovery, and we're members on various consortia where we participate in setting methodologies for example, predictive modeling and so on. So this is the concept of biomarker discovery from our perspective. So we take some high throughput molecular data, this may be genomics, transcriptomics I've already touched upon, and we basically apply either some statistical analysis, a pathway or network-based analysis, or a combination of both to these data to help us achieve certain goals. For example, discovering trends in the data, identifying signatures for patient stratification, predicting response to therapeutics, and so on. And I suppose the ultimate goal of this is to develop some kind of tool, for example, a companion diagnostic that allows for the separation of patients into groups, depending on how likely they are to respond to a certain treatment. So in this simple example here, we would profile a patient using one of these technologies, feed this data into some kind of diagnostic tool, stratification tool, and this tool would then output some kind of decision which can be used in the clinic. So it would guide us as to whether that patient is more likely to respond or not. If they're not, we can then think about other treatments. Unfortunately, this is easier said than done. So there are certain challenges facing us with the amount of data that's being generated at the moment. So as an example, the price of whole genome sequencing will soon be below, if not already below, this magic $1,000 mark. And as you can see by this figure here, after we completed the Human Genome Project, the cost of whole genome sequencing was kind of declining at a rate similar to Moore's law. So Moore's law predicted the number of transistors in a circuit to more or less double every 2 years, and we see this rate was pretty similar with the cost of sequencing. But after the introduction of next generation sequencers around 2007, we see the cost is dropping substantially. So this is generating various challenges because we're obviously generating huge amounts of data. So these data are becoming larger, they're becoming more complex. We face what's known as recursive dimensionality. So this is where we see a small number of samples or patients being profiled and huge numbers of features, so huge numbers of genes or so on. So what we need to be able to do is really start to think about integrating various types of data from these different platforms. We need to make the most of these new technology profiles, such as next generation sequencing for biomarker discovery. We want to be able to reduce the complexity of these huge data sets. We want to be able to distill out the noise from the actual biology, enable us to elucidate some kind of biological meaning. And ultimately, I think everybody's aim really is to start to translate these in silico discoveries into the clinic. So this slide here just highlights various characteristics that we think about when we're looking at a biomarker, and we want the biomarker to have these specific characteristics. So we have classic statistical metrics such as accuracy, sensitivity, precision, and so on. And these really just tell us something about how well that biomarker discriminates between groups or phenotypes of interest. So these are obviously very important. Other metrics that we consider to be important within our team that are often forgotten about are robustness. So this tells us something about how reproducible that biomarker is across multiple cohorts of patients. And during the presentation, I'll show you how we use pathway information to help us improve upon this. And thirdly, we'd like to know something about the underlying biology. So how can we interpret our set of biomarkers biologically? Do they make sense? And all of these components are important to us when we're analyzing these types of large data. So I think to generalize, there are two main approaches for biomarker discovery from omics type data. So we have statistical approaches and we have pathway or network-based approaches. So statistical approaches are usually purely data-driven. So they rely on just that information within the data itself. And pathway approaches often combine these data with some prior knowledge. And this seems to be an up-and-coming trend, and this is where our primary focus lies. And we do actually use both types of approaches at Thomson Reuters within the group. And for pathway-based approaches in particular, we think we've got a strong advantage because the content that we have within Metabase, which I'll talk a little bit more about later, enables us to exploit these methods more effectively. So that was a general introduction into biomarkers. I'd like to actually give an overview of some of the important steps in biomarker identification itself. So this is quite a busy slide, so I'll just take you through it step by step. So we start off with some data, such as microarray data, and this is usually combined with some clinical or phenotypic data, which provides us with some information about the different patient groups of interest. We then perform biomarker discovery using our favorite approaches. I'll discuss some of the approaches that we use later in the talk. And at this step, really, we're able to assess the performance of those biomarkers that we identify. And the next step is what we call model selection. So here we want to select the best model or the best signature after various considerations. Some of those are statistical, like I discussed earlier, accuracy, sensitivity, and so on. And we have others as well. So we might want to consider whether the performance of that biomarker is better than what we would expect by random chance statistically. So in these large data sets, many of the features that we're thinking about may be correlated to that endpoint or that patient group of interest purely by chance. So we can test this by using various techniques such as permutation testing, where we basically permute the endpoints or the groups of patients. We randomly shuffle them many times so that we can calculate the significance of our true performance against some null distribution. So another consideration that we might want to think about is the clinical utility of that biomarker or signature. So incorporation of the biomarker into the clinic is obviously going to be more beneficial if it outperforms or adds some predictive power to the existing prognostic tests. So once a suitable list of biomarkers have been identified, then the next step really is interpretation and validation. So it's back to the laboratory for feasibility studies, where we can think about proof of concept and proof of principle. And ultimately, what we want to do is we have a biomarker which we can measure, and it enables us to separate patients based upon various groups of interest, such as a response to a therapy. So the presentation today will focus mainly on the top half of this, how we find some interesting sets of biomarkers for this downstream validation. So I'd just like to briefly introduce the concept of exploratory analysis before we move on to classification and actual signature generation itself. So exploratory analysis is usually what we term an unsupervised process. So this means that we don't really know anything about the groups within the data upfront, and we want to explore the structure of that data to determine if there are any global patterns or trends within that data that may be interesting. So it also helps us to identify any groups which were previously undefined. So one popular technique used here is something called principal components analysis, or PCA. So this method really just helps us reveal the structure of the data in a way that best explains the variance. So we can reduce the dimensionality or the complexity of that data and enable some visualization of this large feature space in a much more manageable way. And visualizing the data this way may help uncover subgroups of patients which share similar genetic profiles, for example. Another popular method for this type of data exploration is hierarchical cluster analysis. So here, basically, samples are assigned to clusters based on various similarity measures. So samples closer to one another in the dendrogram, which is this image up here, samples closer to one another here contain more similar profiles. And this image here is a famous example where this was successfully used in the identification of various breast cancer subtypes based upon the expression of their genes. So that was just a quick background into some exploratory analysis, but really I just want to focus on classification in today's talk. So this slide just highlights the basic principles. So in machine learning, we want to identify which group a new observation belongs to based upon some kind of training set of data where we already know their class labels, or we already know which phenotype they belong to, and this is classification. So in this example here, we have a group of patients which have been profiled using microarrays, so we're measuring the expression of a bunch of genes, and the patients have a set of explanatory features, and that's their genes. So we use some kind of classification algorithm which basically maps this input data, the gene expression, onto the correct categories, such as disease type 1 and disease type 2 in this example here. This can then become a model in which we can predict the category of the new observation based upon what we saw in the known data. So this is what we call supervised learning, since we already know the correct group that the training set belongs to. So our trained model then becomes a classifier, and this is essentially a mathematical model which contains the classification rule which enables the prediction of new observations. So this classification rule could be, in this simple example, high expression of gene 2 and high expression of gene 1 equals class 1, and low expression of both genes equals class 2. And that's what I mean by classification. So just to summarize, classification is a form of supervised learning. It requires known class labels and learns from a training set to classify new data. We can then essentially assign the probability of a new observation belonging to a particular phenotype or group of interest. We use so-called intelligent algorithms which recognize very skillfully the patterns in the data. So they require a set of predictor variables, for example, genes, and some response variable or category, such as healthy versus diseased. And since the sample space contains large numbers of features, many of these are actually irrelevant. So what we want to do is separate the relevant features or genes from the irrelevant features, the noise. And those relevant features then become our biomarkers or our biomarker signature. So how do we know which are those relevant features and which aren't? So we can use various metrics to assess the performance of those biomarkers. The simplest is probably accuracy, and this basically just tells us how many of those samples we've correctly predicted overall. Other metrics which are commonly used are sensitivity and specificity. So this gives us information regarding the proportion of correct positive or correct negative predictions. So I'll use some of these terms later in the talk, so I just thought I'd explain them up front. Another metric which I might mention is something known as the AUC, or the area under the receiver operating characteristic curve. So this basically estimates the performance of a classifier as we vary the threshold for positive versus negative. And this is a value between 0 and 1, where 1 would be a perfect classification and 0.5 would be kind of random. So before I go into some of the methods, I just want to mention cross-validation, because when we do calculate this performance, we must do it the correct way. And cross-validation really just helps us estimate how that classifier will perform or generalize to independent data, so new data that wasn't used in that modeling process. So the goal here is really just to define a subset of data to test that model against. And one simple method is known as k-fold cross-validation. So the original sample is basically randomly split into k equal sizes. So k is often 5 or 10. And one subsample is used to test the model, and then the remaining samples, k minus 1, are used to train the model. So we repeat this process k times so that each subset is used exactly once as the test set. And then we can take the results from all of those test sets to produce an estimate of performance of how that model or signature will perform when we introduce new data to the system. This slide just gives an overall summary of that process. So here we use 10 repeats of 5-fold cross-validation, which has been recommended in various studies. And basically what we do is we incorporate the whole signature generation process within this cross-validation. So this makes sure that we test the final signature on independent test data each time. So we ensure that we're not leaking information into that training process. And this helps us estimate performance in an unbiased fashion. Okay, so that was just a brief introduction into classification and the concepts, just to give you some background in case it's not something you're familiar with, and help you understand the concepts for when we go to using specific methods and identifying signatures. So I'd just like to give a couple of examples of where biomarker discovery has been successful using just gene level information. And then I'll go on to talk about how we incorporate pathway information to help us further with that process. So this is a famous paper published in Nature in 2002. So what they did is they profiled lymph node negative breast cancer patients, and they were able to identify a 70 gene signature that was predictive of disease-free and overall survival in those patients. And they showed that this signature was able to outperform what's known as the St. Gallen criteria for treatment selection. So this is some criteria which guides therapy selection, basically. And this 70 gene signature actually went on to become the first FDA-cleared microarray-based diagnostic. This is known as MammaPrint, and it's commercially available through Agendia. So that was one success story. Another success story was another example which was published around the same time, perhaps shortly after, in the New England Journal of Medicine. So actually this was a 21 gene signature in the pan-level PCR assay. So this wasn't microarray, this is a different technology used to measure the expression of genes. And this signature was actually able to group, again, breast cancer patients into various risk groups, low, intermediate, and high, according to some kind of tumor recurrence. And this was also shown to be very predictive of treatment effect. So they could basically predict whether patients would benefit from various therapies or not. And as with the previous test, this was also commercialized, and this is now known as Oncotype DX by Genomic Health, and this is also available to use as a guide for therapy decisions. However, from the hundreds, possibly thousands of publications which have focused on gene signatures, there have only been a few success stories, and there are some concerns as to why this is. So in an effort to address this, the FDA led this effort to develop good practices in biomarker signature development, and this was known as MAQC2. This was published in 2010 in Nature Biotechnology. And this paper really compared the performance of multiple analysis teams in generating signatures for a variety of different endpoints. Some of those cancer endpoints, some of those non-cancer, some actually just random. So many thousands of models were developed, about over 30,000 models if I remember correctly, and they generated over 250 gene signatures across the different research groups. And what this study essentially showed was that over a number of different endpoints, no model from the different data analysis teams really seemed to outperform the others significantly. So that gene signature performance for a number of these complex endpoints, for example, prediction of overall survival in multiple myeloma, was unfortunately very poor. And when they measured the robustness of the signatures with respect to the number of overlapping genes across these different signatures, this was also quite low. So it suggested huge variability when using just gene expression data alone. Importantly, when the signatures were actually tested on independent test data, the performance actually dropped. So this suggests that the performance or the biomarkers were not really reproducible across different cohorts using just gene level information alone. And there have been various publications highlighting problems associated with gene expression signatures. So they're highlighted frequently in the literature, and studies are actually showing that random gene signatures are outperforming breast cancer signatures. So, for example, this paper here, they looked at the importance of the 70 gene signature that I discussed. So they assessed the importance of those genes, and they showed that many equally predictive lists could have been produced during that study. So this highlights the fact that the signatures might not really be biologically relevant or candidates for any kind of targeted therapy. And this is really a concern, and questions are being raised as to whether these gene signatures are actually ready for the clinic at the moment. Other studies such as this have compared the reproducibility of gene expression signatures across multiple data sets, and they've again showed that the percentage of overlapping genes which are actually differentially expressed was disappointingly low, 0 in some cases. So again, this just questions the reproducibility of gene level expression data. So there are probably many reasons for this. Some of those are listed on this slide here. So, I mean, we have a lot of heterogeneity in biology, heterogeneity within the samples, a lot of genetic heterogeneity amongst patients, especially in diseases such as cancer. So different variable selection methods, different algorithms, different statistical techniques usually seem to lead to different solutions with similar performance. So we have different lists of biomarkers which perform quite similar. So how do we know which one to use? Gene markers often show poor concordance between those signatures, and importantly, they perform pretty poorly on additional cohorts of patients. So again, it seems that these gene level expression data seem to vary a lot between different cohorts of patients. So I think one of the main reasons for this is because we have such large dimensionality. We have these huge data sets that inevitably we have a large number of false positives that are being identified. False positives are genes which would be correlated with that endpoint of interest purely by chance alone, purely because there's just so many features within that data set. So that was just a general introduction into classification and some examples of where biomarker discovery has been successful using purely data-driven approaches. And I also just briefly mentioned some of the problems associated with that. So what I want to do for the remainder of the talk is really just spend time reviewing the literature and reviewing some of our own research and show you some examples of how pathway and network information can be used to overcome some of these problems and hopefully lead to more robust or more reproducible gene signatures. So as I said, we're seeing this trend towards utilizing this pathway or network information. So that's really just combining these powerful statistical approaches with this prior knowledge in some kind of integrated approach. This prior knowledge is coming from basically databases of signaling pathways, protein-protein interactions, and those types of things. And here I've just highlighted a couple of examples of where groups have shown that incorporating pathway information into the disease classification process actually makes sense, and it does work. So as I said, what I want to do is actually go through some of these examples in more detail and show exactly how we can achieve this integration and how we can show that it leads to more robust results. And it's not just the performance we're thinking about, we're also starting to think about, do those signatures actually explain some biological mechanisms of interest? So before I go into the methods, I just want to talk a little bit about Metabase, which is our database of protein-protein interactions. So all of these methods that I'm going to talk about really rely on some well-annotated network. So we have what we believe to be the most robust source of information within Metabase itself, and we use that as a starting point to all our statistical analyses for biomarker discovery. So Metabase is essentially a database of manually curated interactions. They show mechanism such as binding in this example here between EGF and EGF receptor. They show directionality, and they show some kind of effect such as activation or inhibition. So all of this is manually curated, and this evidence comes from small-scale experiments which has been published in peer-reviewed journals. So everything is referenced. In this case here, with this interaction, there's around 150 publications which support this interaction. So we're confident that these interactions exist, and these go on to form our network. So in total, we have about 1.2 million interactions within our network. About half of these are protein-protein interactions, and the remainder are other types of interactions. We can then put these into some biological context through manual creation of known or well-defined signaling pathways. So we have over 1,000 proprietary pathway maps. We also have a number of disease-specific maps, which have been formed through our what we call meta-minor partnerships with academia and industry collaboration. So we have normal pathway maps, and we also have disease-specific pathway maps for diseases such as cancer. We also have other well-annotated ontologies which we use in our analysis. We have also the public ontologies such as gene ontology. So I think the general idea of using this type of information really stems from the principle of biological modularity. So we know that cellular functions are performed by groups of proteins that work somehow in concert. So if we have individuals with the same phenotype, they may actually show variability in the specific genes that are altered. So this is probably what we see in gene expression data. We have different sets of genes which are, for example, upregulated in patients with the same disease. So there's not a lot of consistency. There's some variability, which is why we don't get this reproducibility across different experiments. However, what we do see is when we map the different genes that are expressed in these patients from the same phenotype onto pathways, they appear to harbor a more consistent pattern. So even though they may not share the same differentially expressed genes, they may share the same dysregulated pathways. This is why we believe that pathway level information is more robust than just using single genes alone. So this slide here is just a high level overview of how this works in the context of analyzing big molecular data. So in a classic data-driven approach, one would typically take some molecular data resulting from the profiling of patients or patient groups of interest. So this data is then subjected to some statistical approach, and we identify some biomarkers using just that data. So the major difference between this data-driven approach and the pathway or the network-based approaches is that firstly what we do is we actually transform the gene-level data into what we call pathway or subnetwork profiles using some high-quality interaction data, such as Metabase. And it's then this transformed data that we use as features in our classification pipelines. And it's then these pathways or these networks which actually go on to become our biomarkers. So I now want to just go over several examples of where these types of approaches have been used and just explain to you exactly how we can use this type of information to further our understanding and to identify more robust gene signatures. So as a group at Thomson Reuters, we've got experience in using these types of approaches. We've been doing this for a number of years now, and I'll begin with an example from our own group. So here, what was done is we took the content of our proprietary maps, and we showed that using actually individual maps as features led to high accuracies in the discrimination of toxic from non-toxic compounds in this case. And you can see here we had high accuracies using just individual maps, high specificities and sensitivities. And in fact, the highest ranked pathway, which is shown here, was actually biologically relevant to the phenotype under study. And it actually performed as well as some gene-based machine learning classifiers, even when used as just a single feature in that biomarker signature. So what we showed was that actually using this pathway information does help, and it does help us gain performance as well as biological interpretation. Another one, an example from Triadica's lab at UC San Diego, uses a pathway activity classification approach. So this is essentially a protein network-based approach for identification of biomarkers using protein interaction networks. So here, actually, biomarkers are represented as what they call condition-responsive genes within pathways. So rather than use all the genes in each pathway and then summarizing their expression somehow, a subset of member genes are identified whose expression levels are discriminative of the groups of interest. So we basically take each pathway one by one and we find out which subset of genes within that pathway best explain or best enable us to classify between the different groups, such as diseased and healthy. So the average expression of this subset of genes then represents the activity of that pathway and then becomes a feature. More than a single gene feature, we have a bunch of genes as a pathway. So then the best combination of these pathways or the activity of those pathways that maximize performance using a classifier are then used as a signature itself. So what they showed in this paper is using this pathway data to identify these subset of genes within the pathways and then building them together into a classifier actually resulted in better performance across independent cohorts than using gene-level expression alone. And particularly when you identify a signature on one data set and apply it to a second data set, the evidence was that using this type of information was more reproducible. This is another example from the literature which actually summarizes genes based upon their scores with respect to being mostly associated with statistically enriched pathways between two different conditions. So what they do is they basically look at which pathways are most enriched, which genes are most discriminative within those pathways, and then use those genes as signature genes to discriminate between cancer subtypes. And what they showed in this paper was that the selected biomarker genes were, first of all, more reproducible across several data sets as compared to using gene-level information alone. So we have a 40% reproducibility compared to 20% reproducibility using single genes. So the performance was there in terms of reproducibility, and also when we look at the accuracy of the discrimination between the cancer subtypes, the signatures were more accurate than using gene information alone. So here we have another example which basically again estimates pathway activity by summarizing the expression level of the member genes within a pathway. So this method actually uses probabilistic inference. So it computes what's known as the log likelihood ratio between different phenotypes based on the gene expression. So we can compute the likelihood of a gene belonging to a particular phenotype based on the distribution across all those different samples. We then combine the scores of the pathway member genes, which represent the pathway level expression. And when we summarize gene level expression in this way, the paper showed that you can then use this summarized data in a logistic regression model. So this is a classifier, and when they used it in that scenario, there was a consistent improvement in predictive performance as measured by the AUC, this area under the curve, which I mentioned before. So there were two main conclusions. When we take pathway information and we develop a signature on one data set, the pathway approach outperforms significantly the gene level approach. So the pathway approach is shown here in turquoise, and the gene classifier is shown in orange here. So you see there's a significant improvement. And secondly, and perhaps more importantly, when a signature was developed on one data set and then validated on a second data set, the pathway-based approach actually retained its performance. So it was very reproducible. So the signature still worked on independent data. However, the gene level approach didn't perform much better than random, as you can see here in orange. So the score is essentially 0. So this is something we commonly see. Gene level signatures aren't reproducible across multiple data sets, and sometimes we're actually just identifying random sets of genes. So something slightly different, but the concept is similar. So in this example, rather than actually use whole signaling pathways, this approach utilizes what are termed subnetworks. So in this scenario, a subnetwork is a small group of genes which are connected directly through known protein-protein interactions. Several of those are shown on the slide. So basically what we do is, for each gene in the data set, we perform what's known as a greedy search. So we combine genes together with other interacting genes within their network neighborhood, so within their interacting partners. So we combine genes in a way so that their combined expression maximizes some discrimination score, some accuracy classifying between groups, or in this case, we can actually use this type of method to predict something like time to an event, so time to disease progression in leukemia, as was the case with this paper here. So this actually enables us to discover subnetworks that best correlate with that phenotype of interest. What the authors did here was found that this bunch of subnetworks they identified were actually biologically relevant, first of all, to the cancer processes that they were studying. And when you actually look at the expression of those subnetworks, they were able to cluster patients into groups based upon their predicted disease progression. So depending on the expression of these subnetworks, patients show very different characteristics in terms of disease progression. And in independent cohorts, these clusterings also retain some statistical significance. So when they applied these subnetworks to independent data, so looked at the expression of these subnetworks in additional cohorts, they still saw the same types of patterns. but when they looked at the gene level information alone, they didn't see the same patterns again. So again, just showing that the reproducibility using this prior knowledge as well as the statistical approach seems to hold better. So here's another example. This is a process known as recursive feature elimination, or RFE. So this is a popular method in variable selection, essentially. So really, feature selection is wrapped around the classification procedure so that we can select which features or biomarkers genes are the most important. So basically what we do is at each step, so we start with all the features in a model. So we start with, for example, 20,000 genes, and we generate a model, and we look at the importance of each of those genes. Those importance can be reflected by some kind of weight. So unimportant genes, so those with low weight, can then be removed at each step. So it's an iterative process. We keep repeating this process until we are just left with an optimal subset of features or genes. So that's standard RFE, but in this method, what they did actually is they used they adjusted the weights of the genes depending on their relationship with one another in a protein-protein interaction network. So, for example, if a gene with a low fold change or wasn't really differentially expressed between different groups, if it had known interactions with a number of other important genes, then it should have some increased influence on the classifier. And this may be the case for something like a transcription factor. So sometimes these only really show small changes at the gene expression level, but they may be key regulators in causing considerable expression changes of many other genes. So in principle, these key genes should be considered as important and correlated to the phenotype under study because they're highly connected to many other differentially expressed genes. So what they do within this study is they basically re-rank the weights based upon an algorithm analogous to Google's PageRank algorithm. It's called GeneRank. So PageRank is basically based on the premise that a web page should be highly ranked in your search results if it's if other highly ranked pages contain links to it, basically. So I think this idea extends naturally to gene expression data, where we want genes to be more highly ranked if they contain links or interactions to other important genes, even if their natural position is lower. So yeah, so essentially the conclusions of this study was incorporating this network topology into the biomarker discovery process improve the predictive performance substantially over the gene level models themselves. And they looked at a lot of different data sets, and they found this to be true in all cases, basically. And the network-based signatures resulted in better performance. So that's just another example where it's been proven that using this type of information actually does seem to help. So here we have a slightly different approach, which uses actually one of Thomson Reuters systems biology tools known as Metacore. And they use Metacore to develop a classifier for disease diagnosis. So one of the focuses here actually is to identify smaller subsets of genes which may increase the clinical applicability of any biomarker signatures that are discovered. So the first step is to identify some differentially expressed genes between the groups of interest. And then we can use Metacore to generate a network representation of that data. So identify candidate genes as those which appear to be in gene hubs. So a gene hub is basically a gene which has large numbers of connections or interactions within a network to other differentially expressed or important genes. We then take just those genes, for example, upregulated, downregulated, and a combination of both in some kind of classifier, and then use those to measure the predictive performance. And again, what they showed is using this type of approach, incorporating this network information, led to better performance than using just that gene level information. So in the interest of time, I think what I'll do is I'll move on to some preliminary results from the Orion program. So basically, so Lenny actually already gave you a brief introduction to what this is. So in a nutshell, this program aims to take a systems biology approach to furthering current understanding of brain diseases. And this is really a challenging area for research at the moment, especially when trying to predict which patients will and will not respond to treatments. So the first phase of the Orion program focuses on multiple sclerosis. And just a brief introduction into the program before I go on to some of the preliminary results. Really, the program aims to bring together large data sets with multiple partners. And the aim is really to integrate high dimensional data. So this has an abundance of clinical and phenotypic data to it. And we're using predictive analytics to generate what we call biomodels that represent disease processes. And it really is a truly multidisciplinary effort with many aspects. So there are data modeling partners who integrate this diverse data and build predictive disease simulations, partners dedicated to providing real-time disease data, long-term patient data from clinical studies. And as you can imagine, this generates a huge amount of data. Importantly, the models which we generate in this alliance are going to be disseminated to the public, and hopefully this will assist in developing strategies for biomarker-driven clinical trials in the future. So we, Thomson Reuters, were chosen to be one of the computational modeling partners in this program. And what we've been doing is we've been utilizing our systems biology platforms, some of the methods I've talked about today, to really analyze these big data with the aim of identifying disease subtypes, biomarkers, signatures, and so on, which help tell us something about disease progression in multiple sclerosis. So this again is quite a busy slide, but essentially what we're doing is we're leveraging our pathway and network content to identify pathway and network-based signatures, which enable patient stratification. So they help us uncover new subtypes of disease using, as I said, some of those approaches that I've talked about today. So I can't go into too many details, but the results are very preliminary, but they do look quite exciting. So what we've been able to do is basically take that mRNA data, that gene expression data, for 200 or so patients, and identify several important pathways and gene subnetworks whose expression profiles seem to be associated with time to relapse in this disease. So what we've done is we've looked at the actual activity of those pathways across those different patients. And this really is just a heat map which shows the expression or the activity of those pathways in the rows, and each patient is in one column of this heat map. And what we see is that the patients clustered according to the activity of these biomarkers. So we see 4 main clusters of patients within this heat map. They're shown here in blue, in green, in orange, and in red. So you can see the expression of those biomarkers seem to change quite drastically depending on which cluster that patient belongs to. And what we went on to do was actually show a survival model where we can actually visualize the points at which patients develop a relapse over time in this cohort. And interestingly, the patients had very different relapse times depending on which group they belong to. So for example, in cluster 1, here in orange, this represents a very good prognosis group. So there weren't many relapses within this patient group here based upon the expression of these pathways for this specific pattern. Whilst cluster 4 in blue, this was a very poor prognosis group. So this was a small group of patients who actually all developed relapse very quickly. And this group was actually about 17 times more likely to suffer a relapse at any given time point than cluster 1. Cluster 2 here in red appears to be some kind of mean profile across the population. So we've gone on to investigate the genes involved within these pathways, and what we're seeing is many known disease biomarkers and drug targets, as well as unearthing some previously unknown gene disease associations. So we see a lot more MS biomarkers or drug targets within our list of biomarkers than we'd expect to see by chance alone. So that's potentially very interesting. And additionally, if we look at other neurological disorders, many of the identified biomarkers in MS are also biomarkers and drug targets of other conditions such as schizophrenia, dementia, depression, and so on. This shows that we have a significant amount of overlapping biology across these diseases, which is potentially worth pursuing. Okay, so just to conclude, so that was really a whistle-stop tour of everything we're doing at the moment, and there was a lot of information. So just to summarize, biomarkers are molecules indicative of normal or abnormal processes. So there are many challenges in analyzing these complex molecular data. Statistical techniques such as classification help us to identify biomarkers which represent specific groups of interest. Gene expression biomarkers are often not reproducible or robust across data sets. So incorporating prior knowledge in the form of pathways or networks may help us improve upon this robustness and also may help us explain the underlying biology better. And these types of approaches are being used successfully so far to further understand diseases such as multiple sclerosis within the Orion program. Okay, so yeah, so that's the end of my slideshow. Thanks for your attention. And let's see if there are any questions.
Yes, Lee, if you on the Q&A app, we do have one question from the audience right now. I just want to ask the audience, on the lower right-hand side of your screen, there is an Ask a Question button, and so you can certainly click that and submit a question to Lee. I did have a question myself, Lee, of a lot of the diagnostic, or I'm thinking specifically of oncology diagnostic companies that are out there. The majority of them are gene-based. Are you starting to see them adopt the pathway approach to provide more accurate diagnostic tools in the future?
In the actual commercial diagnostics themselves?
Yes.
So I don't think it's something which are available at the moment, but I think there seems to be some effort to move towards this, certainly. So a lot of the publications are now starting to incorporate this information because it's becoming more and more reliable. We have a wealth of it. So I think people are becoming more aware of the fact that they should consider this when they're analyzing their data. So I think we'll get there eventually.
Great, thank you.
So there's a question, do you have any experience with biotheranostic cancer type? Unfortunately, I don't, so I can't really answer that, I'm afraid. So it's not something I'm familiar with or we've come across really. But if there are any other questions, please go ahead.
Yep, looks like one more question came in.
Okay, let me just see if it pops up. Okay, so the question is, would you say a bit more about the random relabeling testing that you used? So yeah, sure. So basically, there's something called permutation testing, which enables us to assess if our signature is better than or performs better than what we'd expect by chance. So because these data sets are so large, we can often find features within them which are related to the endpoint or the class labels purely by chance. So we can do various things, and one of those is permutation testing. So we take our signature and we have some kind of performance on the real endpoint. So we have a classification performance. Then what we can do is we can shuffle the class labels of that data. So we can randomize which patients are diseased and which are healthy, for example, and then retest the performance of that signature. And that gives us some kind of idea as to how well that signature will perform on some random data. We repeat that test many thousands of times, and that helps us create some kind of null distribution as to how well that signature will perform on purely random data. And then what we would hope to see is that the real performance of that signature is above the performance that we see on that random data. So it allows us just to estimate some kind of confidence as to how well our signature is performing, and we can give a p-value then. So that's something we did in the Orion program. So we have this list of pathways, for example, which are associated with the endpoint, and then we shuffle the patient's endpoint data. So we shuffle the times it took them to develop a relapse, for example, and we repeat that many thousands of times. So we have a p-value then for each pathway as to how significant it is associated with the time to relapse in those patients. So hopefully that answered your question.
We are coming up on the hour. Are there any more questions, Jeremy?
No, not that I see. Let's see if anyone else in the audience wants to ask a question. Please select that Ask a Question button at the lower right of your screen. Other than that, if nothing else comes in, I'll turn it over to you, Lenny.
All right, well, I will just give a little plug for our next Orion webinar. First, thanks to Lee Lancashire. Thank you very much, Lee, for doing that. I'm sorry, I forgot to mention that you've been shortlisted for a Booker 5 times and you won the Tour de France twice, correct? Is that correct?
It's correct, but I try not to brag about it.
Okay, well, I'm sorry. I forgot those when I introduced you. But thank you very much. We'll look forward to the next time. On July 23rd, Stephen Larson, PhD, the CEO of Metacell, will be delivering the webinar titled Collaborative Bottom-Up Modeling in Computational Biomedicine. We'll look forward to that. You'll all be receiving reminders in the mail. Pass them along to all your friends. And for now, Orion Bionetworks TV is signing off. Thanks for joining us.